Alright, in this data video we're going to be looking at how to do a method, because a lot of students get confused when it comes to method because they feel like they just need to recount what they're doing. Oh yeah, we did this, we did that, we mixed this, and then we recorded our results. But unfortunately, in a scientific method we need to look out for some key features and ideas so that it allows other scientists to do the experiment, to replicate the experiment in a scientific way. So let's have a look. Well, a method really is just like you would bake a cake, right? So the whole point in trying to bake a cake, right, is you need to follow a set of procedures. You need to make sure that you've got the right equipment, that you've got the right materials, and that you're following the right steps. And hopefully the cakes that you see in the picture will turn out to be the cakes that come out of your oven, right? Pretty much close to as much as possible. Well, certainly not in my oven, but there's more skill to it than just following recipes. But that's the whole purpose, is that you're told exactly what to do, when to do it, what materials to have, and then hopefully you should be able to replicate the baking process. Well, the same thing is for scientific reports. We need to do an experiment, but if I wanted someone to do an experiment overseas and try and test it to see if they get the same results as me, they need to know exactly what did I do, how did I do it, etc., so they can replicate it and see if they get the same results. I mean, the laws of the universe and chemistry are consistent, so if they do it the same way that I did it, you hope that they should get the same results. So when we do an investigation, we want it to be in an objective manner, right? So objective means that you're not imparting any of your own bias into it, right? We don't want to somehow skew the results. We want to be as objective as possible. We want it to be repeatable and reliable in every way. So when we do an experiment, we know that it's not due to chance. So when we measure the boiling point of water at sea level and we do that in the same conditions, then we should have the same value for the boiling point of seawater. It just shouldn't be the chance of that boiling point. And so in order to do that, of course, we have to make sure that our variables, etc., are kept constant. And we did another video previously on what are the different variables. But once we know what our variables are, we've got to write our method so we know how to do the experiment. So let's look at the key features to watch out for with a good method. Now, I've got a recipe here that can be found at this address here, so thank you very much for that recipe, but it really illustrates some of the basic ideas that you need to have in a scientific method. I mean, here when we're making the soup, it tells you exactly what you need to do. Now, I notice that it says the amounts, the quantity. We need to have half an onion diced. It's not just saying, get some onion, get some carrots, get some potato, maybe some celery, some green beans, get some zucchini. It's not just telling you to get it, it's telling you how much to get it. But not only that, it's saying that the onion should be diced. So it's giving you more information. So when we do a scientific report, we don't just say, get some hydrochloric acid. We will say, get 1 molar hydrochloric acid, and we will even specify how much you need. 10 mils of 1 molar hydrochloric acid. So we're being very specific that we've got to make sure that's in our method there, right? So that's our materials, right? What else do we know? Well, not only do we have the materials in terms of food, we've also got other things that we need as well that's coming into there. So we're saying things like, we need, it says a tablespoon, so we need some way to measure. Okay, we don't use tablespoons in science, we use other measuring equipment, right? So measuring cylinders will tell us what we need to measure the volumes, we need electronic balances. But here they're saying we need a tablespoon, so that's their measuring device there, so they can measure the liquid oil. Okay, and so that is very precise. We need to include that. What else do they have? Well, in their actual method, their steps here, their instructions, notice a few key features. They're saying place each vegetable in a separate pile as they'll be cooked at some different times. Heat the oil in a large saucepan and onions and cook them over medium heat until they are transparent. So it's giving you some instructions. It's not just saying get some onions, put them in a pot and heat them. They're telling you roughly how long to heat them for by saying heat them. They're telling you roughly what temperature to heat them, medium heat. And they're saying, well, you heat them until something happens. In here they're saying until they're transparent. So they're saying information in their method. And we've got to put that in our scientific method. We're going to tell people how long to do things for, how much to add. Okay? And so be wary there. Now, see here they're saying add carrot and cook for 1 to 2 minutes. Now notice with all these descriptions here that it's neutral language as well. They're telling you what to do with verbs, so they're saying heat, they're saying add, stir, okay? So we're telling you what to do there. They're not just saying like a recall where some students write, first add some water into a beaker and then we did this. Well, they're not using that type of language. We're using neutral language here. Okay, and so we're going to try and look for these main features and put them into our scientific method. Okay, so let's have a look here. Now, of course, different teachers will do this differently depending upon what year level you're in, whether you're juniors or whether you're seniors. They may become more sophisticated with seniors, but the general idea here is we have to say, what do you need? How do you set it up? And how do you do it? Now, some teachers will just have the word method and then write everything as a series of steps. So they might say, step 1, collect the following pieces of equipment, and they'll name the equipment. Whereas another teacher might say, well, I'm not going to have that as a step. I'm going to tell you exactly what you need before you actually go into using it. Okay. Another teacher will have a separate diagram or section that says, this is how you set it up. Whereas another teacher might put that diagram within the steps. So it depends on what your teacher wants to do, but it's pretty much the same stuff I'm going to show you here, but the format might be slightly different. But I've decided on this format for this video here that we're going to start off with our method is going to be containing the first section, which is equipment and materials. So that's telling you exactly what you need to collect. So it's very similar to what we've got here, right? Saying this is what you need to get. You need to get all your onions prepared. You need to get all your diced tomatoes. You need to have all your measuring equipment ready to go, because once we start cooking, you need to have it there. You can't just go, oh, I need to go find something now. So they've got a separate section to tell you what you need. Well, in our method, our first part of our method is equipment and materials. Now, I haven't just called it equipment because I'm also telling you to get some acid here as well, right? And of course, I've just noticed a typo there. It should be hydrochloric acid, right? Not hydrochloride acid, right? So make sure it's hydrochloric acid. So what I'm doing here is I'm trying to tell you exactly what you need. You need to know 2, and we've got a volume. So I need to go collect 2 250 ml beakers. I'm not saying just go get some beakers, okay? And I've got 250 mils, that's the volume I need, and I need to make sure it's 1 molar hydrochloric acid. So make sure you've got your very specific, right? And I'm just going to make this wider. I'm being very specific with amounts, okay? I'm being quantitative. I'm telling you exactly how many I need. Then my next section here is the experimental setup. So we can draw our experimental setup so it's very clear to the people doing the experiment how you're going to set it up. Now remember, of course, we've got to ensure that our diagrams are fairly large, right? We're not going to be squeezing them into some little small spot on the page. We're going to take up a good section of the page so it's very clear, because if you have lots of labels to add or you've got multiple things to draw in your diagram, let's be doing distillation, and you've got equipment going everywhere and hoses, you don't want to squeeze that into a small space. You want to take up a good amount of space so it's not cluttered. Because if it's not cluttered, it means it's very clear to the person reading it. So I would say look at about 1/3 of a page. Okay, so whatever size your page is, do about 1/3. And of course, we're going to be doing pencil. We're drawing our diagrams with a ruler. And we're using 2D, so we're drawing our equipment as 2D, not 3D. So we can see here I've got 2D drawings, and I'm using the proper drawings for my equipment. It's all in the correct ratios, or the size, or the scale, I guess you could say. Okay, so I've got my diagrams there. I've got it labeled. So not only is it labeled, it's labeled from the side. It hasn't got labels going all over the place like this. That looks like a bit like a spider web. Okay, so let's make sure it's nice and neat. And I should really have a title up here, okay, to tell the person exactly what it is all about. So once I've got that, I then go into my procedure, right? The procedure is how you do the experiment, right? So I've got materials, I've got my chemicals, I've got my diagram, and then now my instructions is the procedure. Okay, so let's go on to the next page here. All right, so here's an example of a procedure. Now, of course, I've only got a small amount of screen space here, so I'm just trying to put in the main aspects. When you do your method, you want to make sure it's detailed. Some methods, depending on the experiment, can be a whole page long or multiple pages, depending upon the level of detail that you want to go into, and of course how complicated the experiment is. So let's look at some key features here I've tried to highlight to show you what to look out for, right? We've already talked about the format. Let's look at our procedure now. So we're into this section here. We want to make sure they're in steps. So let's get the highlighter out, and I've got my steps here. See, step 1, I'm going to make that a bit bigger, step 2. So we want to make sure it's very clear that they're in steps. Okay? Don't just write a whole big paragraph and it's all intertwined and you've got to extract the information. Keep it clear, keep it steps. Step 1, do this. Step 2, do that. Think of this part here as like the instructions you would get on how to build something or how to bake a cake. If I was to tell you to pull apart a motor, I would say, okay, step 1, get this bolt and undo this one, and I'll be telling you at each step of the way. Okay, so now numbered steps. The next thing is I've got neutral language. So notice that everywhere here, if you read some of it, you should not be able to see, and I'm going to change my pink here, right? You should not have things such as I, we, you, they, okay? We are keeping it neutral language. It's not first person, it's not second person, okay? Get rid of all of that, and they're just nice, clear statements that has neutral language. Of course, now, if I change the color again, so let's go to a blue now. I've got verbs in there to tell you exactly what to do. So I've got things like measure. I've got things like pour, okay, record, rinse, pour. So I'm telling you what to do here and simultaneously place. So I'm telling you what to do with verbs, okay? I'm not just saying, you know, put some magnesium into test tube and record the results, okay? I'm being specific, so let's do another color here. So let's do orange now. I'm being specific. Okay, I'm telling you amounts of times and I'm telling you to be descriptive. So I'm saying 10 mils, 5 mils, I'm telling you how much to do. Okay, I'm saying simultaneously, I'm telling you what to do at the same time. Repeat it repeat these steps 3 times. So I'm trying to be very specific what you're doing. If I'm doing something, let's say I said test this gas in this test tube, I don't just say test the gas. What I'll say was be specific and say light the match, place the match over the top of the test tube, then release your thumb. Okay, so I'm trying to be very specific on what you need to do. All right. I want to make sure that I've got clear control variables. So let's do red now. Right. Remember the whole point of when we do our experiment is we want to make sure we've got our controlled variables not changing. We want to make sure they're the same. The only thing that should change would be our independent variable. So in my method, I've got to really have links there to ensure that when you do this method, you're keeping everything the same. Okay, and so I'm saying things like using the same cylinder, right? Measure 10 mils. Right? I'm not just saying measure another 10 mils. I mean, you hope that when they read it, they go, oh yeah, I better use the same measuring cylinder. But I'm saying like rinse. So that way I'm preventing cross-contamination. Okay, because remember in this example, you've got different concentrations of acid. And if I just say measure another 10 mils, you're going to have cross-contamination of the first acid. So I'm saying rinse the measuring cylinder 3 times and then measure 10 mils. Okay, so I'm keeping my control variables in the method there to make sure everything is consistent. I'm also being very clear. Let's get a different color. Let's do green, dark green. What do I record and where do I record it? So rather than say just record your results or see what happens, we've got to be very, very consistent. So record this initial temperature. So that's what I'm recording. Where? In a results table. Okay, and then I'll be saying things like down in step 5, record the time, and where do I record it? In the appropriate column in the results table. So I'm telling you what to do, what to measure, and where to place it. Okay, hopefully your results table's there ready to go, but I'm being very specific. And then of course, for our last sort of thing to watch out for is repeat trials. So to make our method and our experiment more reliable, we want to carry it out more than once. Because if you just do it once and you make an error, you don't know. But if you repeat it 3 or 4 times and you notice something a bit different, you go, hang on a second, this is different to what we got before, then there's probably an error somewhere, and that's going to make you think about your method very carefully. And so that's something we want to do. And so down here I've got repeating it. Okay, so there are some things to watch out for, and when you write a method, make sure it's detailed, make sure it's got all of these features here, and you can't go wrong. Because most of the time when students write a method, it's pretty much, yeah, we did this, we did that, we mixed that, and then we recorded our results. Well, that's fine if they're thinking about it, but if I was to give those set of instructions to another student in a different country and say, off you go, do that, they will go, what do you mean? They need to have specific instructions. So hopefully that makes sense. You've got your notes in your book. You've got these checklists. You've got these checklists maybe written in the side column of your Cornell note-taking method that we always use. And you've got an example, and you've used colour like I have. Right? You've got notes with colour, so it really makes it very clear to your cells, your brain cells, your eyeballs. So it reinforces this. So when you do it with a practical, you can check it. So I'll see you in the next video.